It's not often that one has the honor of introducing someone of his accomplishments, but also someone whom you can honestly say, I knew you win. And this goes both ways for Doug and me. Doug Rees is the Roscoe Gilkey Dixon Professor of Chemistry at the California Institute of Technology, and he is also an investigator of the Howard Hughes Medical Institute. Doug is also Adjunct Professor of Physiology at the UCLA School of Medicine. Dr. Rees is a member of the American Academy of Arts and Sciences, as well as the National Academy of Sciences. His research interests are in the molecular mechanisms of bioenergetic processes using X-ray crystallography and functional approaches. His laboratory studies the structure and function of metalloproteins and membrane proteins, particularly those involved in ATP-dependent transduction processes. Dr. Rees received his Bachelor of Science in Molecular Biophysics and Biochemistry from Yale College in 1974, working with Carol Slayman. He conducted his graduate work in protein crystallography with Bill Lipscomb at Harvard, receiving his PhD in biophysics in 1980. If you read Doug's HHMI bio, he talks about initially avoiding crystallography when he went to Harvard, based on some misconceptions. Because in the mid-70s, there were a limited number of proteins that were naturally present in high abundance that could be studied. And also, there were not a lot of academic jobs available for people studying proteins by crystallography. However, he took a course with Steve Harrison and Don Wiley, which changed his view and his attitude, because understanding the protein folding problem and gaining insights about the protein folding process could be achieved by determining the structures of proteins using X-ray crystallography. After completing a research rotation in Steve Harrison's lab, Doug Rees was hooked. And upon completing postdoctoral work with Harrison and also James Howard at the University of Minnesota, Dr. Rees joined the faculty at UCLA in the Department of Chemistry and Biochemistry, where he was an assistant professor from 1982 to 1986. And it is here that our professional paths crossed, because the Eisenberg, Rees, and Dickerson labs were all on the second floor of the Molecular Biology Institute, which is now Boyer Hall at UCLA. And our research groups, the graduate students, postdocs, and the technicians commingled in the wet labs, the X-ray labs, the computer modeling labs, and the offices, and we held joint research group meetings. Although I worked with David Eisenberg, Doug was also a mentor and a teacher. And I remember Doug without his beard, and he remembers me with my weird Dorothy Hamill wedge haircut. After earning the rank of associate professor with tenure in 1986 through 1989, Doug moved to Caltech as professor, and many of us moved on to further academic careers as well. When I discussed with Doug my move to the California State University, Doug was one of the most supportive of my faculty colleagues. In 1997, I spent my sabbatical in Doug's lab at Caltech, and at that time, Doug shared with me his tremendous admiration and respect for faculty in predominantly undergraduate institutions. We do it all, and we have to do it well. Doug holds teacher scholars and undergraduate research in the highest regard. Another crystallographic familial tie to Doug Rees in this department is Erik Bryan, our staff scientist at CMOLS. Erik's postdoc in Doug's lab from 2007 to 2009. Among his many honors and awards, in addition to memberships in the AAAS and the National Academy, Doug earned the Excellence in Teaching Award from the Associated Students at Caltech. And he also was awarded the Dorothy Crowfoot Hodgkin Award from the Protein Society. Doug currently serves as an associate editor for Protein Science, and he's a member of the proposal review panel for the new LINAC coherent light source at the Stanford Synchrotron Radiation Laboratory. Please join me in welcoming Professor Douglas Rees.
I didn't need to wear glasses back then either. Thank you very much for the kind introduction, and it's a real honor to be able to have the opportunity today to present the Goldstein lecture. I think universities are really wonderful places, and one of the reasons for this is they attract such remarkable people as Chevy Goldstein, and through their teaching and research and mentorship, friendship, and so on, they really end up impacting a large number of people, and so I'm really privileged to be able to have this opportunity today. I will be talking about our work on nitrogen fixation, so just to put this into some context, nitrogen is an essential component of many biological molecules, proteins, nucleic acids, vitamins, also signaling molecules like NO. And even though we're surrounded by this abundant atmospheric reservoir of dinitrogen, most organisms are unable to directly access nitrogen for our own metabolic purposes. Unfortunately, there's a certain class of organisms known as nitrogen-fixing bacteria that are able to convert nitrogen in the atmosphere to a metabolically usable form of ammonia. Nitrogen fixation represents one facet of the nitrogen cycle that involves the global interconversion of nitrogen through all these different oxidation states. Starting, say, with atmospheric dinitrogen, 78 or whatever percent of the air around us, nitrogen-fixing bacteria, either free-living or in symbiotic relationships with various plants, are able to convert nitrogen to the reduced form of ammonia, which then gets utilized further by other bacteria in the soil who, just like we use reduced compounds to generate energy, oxidizing them, bacteria are able to take ammonia to nitrite to nitrate and use this to drive their energy metabolisms. And ultimately, nitrates get returned to dinitrogen through the process of denitrifying bacteria. Because of this process of denitrification, eventually all the fixed nitrogen in the biosphere would get converted back to N2. And so nitrogen fixation is essential for life, for sustaining life on Earth by replenishing the amount of nitrogen that's available in metabolically usable forms. So it's sort of an example of the consequences of insufficient nitrogen fixation for agriculture and the economy can be seen from a development a little over a century ago that has sort of remarkable parallels to the types of discussions we have today on oil and energy. So Williams Crookes was a British scientist. He developed the Crookes tube, which generates X-rays, which is good, and also this Crookes radiometer, which you may have seen with the vanes that put it in light and it turns around. So he did the analysis following that he looked at the rates of consumption and production of some commodity. In this case, it was wheat, and concluded that by doing a linear extrapolation of the rate at which wheat was being produced and eaten, that wheat-producing soil is unequal to the strain put upon it, and England and all civilized nations stand in deadly peril of not having enough to eat. Okay, and so by this extrapolation, that doomsday scenario was 30 years. Now, Crookes had a sort of what we would say a narrow view of what constituted a civilized nation. It consisted of Europe, the east coast of the United States, not Asia, Africa, or the west coast of the United States. So anyway, Crookes realized at that point the limiting quantity to agricultural production was the amount of nitrogen that was fixed in the soil. And further, it was known at this time that living organisms were responsible for nitrogen fixation. And as he so picturesquely said, the nitrogen with which a light heart we liberate in a battleship broadside, because of course fixed nitrogen is used to make explosives as well, has taken millions of minute organisms patiently working for centuries to win from the atmosphere. Okay, well, so 100 years ago, the main source of fixed nitrogen was not in the Middle East, but it was in Chile, the sodium nitrate or saltpeter deposits. And if you knew the rate at which you needed the fertilizer and the size of these deposits, you could calculate this 30 years for famine. Well, of course, then other people said, you know, we've underestimated the deposits. You know, there's all this great extraction technology and so on, and that these would last for 200 years, and that these earlier estimates were alarmist. Okay, so at the end of this report, Crookes made a statement that I think all chemists have grown to love. It is the chemist who must come to the rescue of the threatened communities. It is through the laboratory that starvation may be turned into plenty by developing a synthetic method to fix nitrogen, basically converting nitrogen in the atmosphere into a form that could be used by plants and organisms. Now, Crookes had this view that the way in which this could be accomplished is by oxidizing nitrogen to nitrates. And this was actually, which is an energetically favorable process, although it has a huge activation energy and requires very high temperatures. And this was used in places with abundant electricity, like hydroelectric dams at Niagara Falls in Norway and so on. But remember, this was in the early 1900s. Within 15 years or 10 years, Haber had developed in Germany a catalytic system for reducing nitrogen directly to ammonia, which was then industrialized by Bosch. So this was a completely different process than Crookes had been envisioning. And then over the last 100 years, this has completely transformed society, in that just because of the enhanced agricultural production, the Earth has supported the population increase of something like 5 billion, mainly because of the ability to fix nitrogen. And just to put things into perspective, each year roughly 1 part in 10 to the 7th, I think it's 10 to the 7th, of atmospheric nitrogen gets converted into fixed nitrogen, which means since this is a cycle, it gets restored. And 100 years ago, biological nitrogen fixation was essentially the only route. There were some contributions from lightning and volcanic emissions. But over the last century, the Haber-Bosch process, it essentially produces a comparable amount of fixed nitrogen each year in the form of fertilizers for agriculture and the chemical industry. And nowadays, of course, we're saying the chemists need to come to the rescue of the threatened communities by developing a method of fixing carbon dioxide, so artificial photosynthesis to reduce CO2 to hydrocarbons, for example. So this discovery had a number of consequences for Haber and Bosch. Actually, the Bosch family is very inventive. So if you ever had a Volkswagen, you'd buy Bosch spark plugs and coffee makers and so on. And so that's part of the Bosch family. Both Haber and Bosch won Nobel Prizes at different times for this work. In Haber's Nobel Prize address, now remember, this was at the end of World War I, and so the development of the Haber-Bosch process was sort of during World War I, and by a number of sort of analyses, probably prolonged World War I by several years, as well as all subsequent wars, because of course the British cut the Germans off from the saltpeter deposits in Chile, and this method allowed the Germans to be able to make fixed nitrogen for agriculture and for explosives. Okay, so in his Nobel address, Haber says this solution is not the final one. Nitrogen bacteria teaches us that nature, with her sophisticated forms of the chemistry of living matter, still understands and utilizes methods which we do not as yet know how to imitate. And so what I'd like to talk about today is at least some progress that we've been trying to make to try to understand how the biological process works. Now, I say Haber was a very complicated person. Not only did he come up with the Haber, at least the Haber part of the Haber-Bosch process, worked with Bosch. He also oversaw the first use of gas warfare that the Germans used against the French. And you can imagine, you know, he didn't win the Nobel Peace Prize, but you could imagine that the French and the British weren't so excited about this. He actually was Jewish, and even though he was a German patriot, ended up finally leaving Germany because he was cut off from his position, and he died a sort of a bitter and broken person. And there's been some fascinating biographies, one recently by Thomas Hager on the alchemy of air, which they call this mankind's greatest invention, because it led to the ability of increased agricultural production to feed all these people. But anyway, really interesting, not only scientifically, but sociologically and so on. Okay, well, our approach to this problem has been